We've all had wounds before, and we've all experienced the wound healing process that comes afterwards firsthand. There's inflammation, there's scarring, and then eventually the wound closes up. But underneath the surface, underneath the skin, lies a much more sophisticated process. On a cellular level, macrophages and fibroblasts are critical to wound healing. They're involved in a variety of processes: inflammation, proliferation, scar formation, and the formal wound closure. These cells communicate with each other by releasing chemical signals that will activate processes within other cells. Now, these cells also communicate with each other through direct contact using gap junctions, which are membrane channels that are made up of proteins called connexins. This form of communication is a little less known, so we wanted to quantify connexin expression. We did this by seeding macrophages and fibroblasts onto well plates. And we seeded the cells individually, but we also seeded macrophages and fibroblasts together, in which they were in direct contact with each other. After that, we performed a qPCR, which quantifies gene expression. And we wanted to do this for connexin expression. After we performed the qPCR, we noticed that connexin expression was increased when the cells were in direct contact with each other. This is especially true for connexin 43 in macrophages. Connexin 43 expression has been shown to increase inflammation. But when connexin 43 expression is reduced, cells increase blood vessel formation. So we wanted to manipulate the connexin 43 protein and examine its effects in wound healing. We did this by performing an siRNA knockdown. In an siRNA knockdown, a double-stranded RNA molecule will enter a protein complex. The passenger strand will be discarded, while the guide strand will be attached to an mRNA strand that codes for connexin 43. Now, this guide strand and mRNA strand will be chopped up, and then its expression is going to be reduced. And this will be the case for connexin 43. In order to perform the siRNA knockdown, we used the lipofectamine reagent. After optimizing its concentration and time, we were able to achieve a transfection efficiency of 90%. This means that 90% of the cells were able to incorporate the lipofectamine reagent. There's one issue with that. Lipofectamine is toxic to the cells, and when we added that reagent, sometimes the cells would die out. So we decided to try out another reagent instead: interferon. When we used interferon, we were able to keep the cells in a healthy state. And in addition to the cell's health, we were also able to maintain the same high transfection efficiency. So we decided to choose interferon instead. And after running a qPCR on interferon, we were able to achieve a knockdown efficiency of about 60%. So this means that 60% of the cells that incorporated the interferon reagent were able to knock down connexin 43 fully and reduce that expression. So now that we have successfully knocked down connexin 43 with interferon, our next step is to examine that effect on wound healing through an assay that will help us detect inflammation. I would like to thank all the people from my lab for their support on this project, and I would also like to thank you all for listening to the project pitch. Thank you very much.